let's see. Hello my dear little champions. I hope I am audible, I am visible. Let me check it by myself also. Yes. So we'll just wait for like 5 seconds, 5 to 10 seconds, and then we'll start off the video for biology's first chapter, which is, as you can see, principles of inheritance and variation, my dear children. And along with that, my hair did the magic of showing you the first question. But we'll just wait for a few seconds. Hi Shagufta. Okay, chalo, let's get started. I see one child, and with one child we are going to begin. Okay, so the first question says, this is a question from the model question paper, so I have given priority to that. What if these questions come on the day of exam as well? Yeah, so I hope we have already did it, but still, if you have it, if you have not, then let's do it. How are you? I am really great. I hope you are also doing really good. Yeah. So the first question says 37% recombinant Drosophila progeny obtained in the T.H. Morgan's dihybrid cross experiment with the phenotypes red eye color, normal body and white eye color, miniature body is due to. So in short, in this question they are saying that there is 37.2% recombination, my dear children, and I'll tell you one thing, 50% is the maximum. And in that, if someone is showing 37.2, that means they are actually showing high amount of recombination. And I'll tell you a story of two best friends. So over here, you and your best friend, if you're too close, you won't allow the entry of the third person over here, right? Similarly, the genes which are there on the chromosome, if they are too close, we call it closely linked genes. Okay, so they are not going to go separated, and if they are not going to get separated, they would not combine with someone else and show something that is known as recombination. So we got to know one thing, that is closely linked genes are not going to show too much amount of recombination. But if they are loosely linked, if they are found somewhere over here and here, then they can show recombination. What do you exactly mean by recombination is that this part of the chromosome can move to some other part of the next chromosome, and hence you will get a new combination of things. That is what you mean by recombination. So if 37.2% recombinant is there, that means there is too much amount of recombination. If there is too much amount of recombination, that means they are not close, they are loosely packed, they are far away from each other. So I have to find something that says that it is loosely linked and longer distances are there between the genes. So over here, if you see the C option, it says that they are loosely linked as in they are far away from each other. That is what you mean. Got it? So that is the first answer. I hope every one of you got it. So whenever the recombination percentage is way too much, just understand that the genes are far away, they are loosely linked. Okay? If the question was like this, they show 1.2% recombination, then you would have to choose the one that are tightly linked, they are close, very close to each other, such that they are not allowing the formation of what? Recombinants. So the answer to this question goes C. Children, you can also keep on guessing. Hi Aman, good evening. Thank you for asking with how I am doing. I am doing really good. Chalo, let's go to the next question. Yeah? This question's answer you can give me in the live chat. Children, I am seeing the live chat over here. I see 9 people over there. So give your answers. I am ready for that. Yeah. So in a dihybrid cross in pea plants, Mendel got 9:3:3:1 phenotypic ratio. It denotes that. So he got a ratio of 9:3:3:1 in a dihybrid cross. What do you mean by dihybrid cross? Where two characters are studied at a time. For example, the shape of the seed, round and wrinkled. Okay, this is one set of character. What is it? Seed shape. And then along with it, one more character is studied, for example, seed color. Now, when he did the experiment, Mendel did the experiment, he got this particular ratio. Why did he actually get this particular ratio? That is a question. It denotes what? The alleles of two genes are interacting with each other. Are the alleles of both the genes? What do you mean by alleles of both the genes? For example, for capital R is for round and small r is for wrinkled. Okay, so these are the two alleles for this particular gene. Now similarly over here for seed color, Y is for yellow and small y is for green. These are the two alleles for this particular trait. Now alleles of two genes are interacting with each other. Obviously they interact, but that does not have to mean that they give a ratio like this. It is a polygenic inheritance, have nothing to do with this question. It is multiple allelic inheritance, again nothing to do with the question. The alleles of two genes are segregating independently. This is the answer. That is, for example, capital R, okay, can go with capital Y. Capital R can also go with small y. The alleles are assorting independently. If you still didn't understand, just listen to me carefully. This is one set of chromosome having capital R, small r. This is another set of chromosome having capital Y, small y. Now you know that when gametes are formed, from one pair one allele goes. Suppose from this pair one allele is going, that is capital R. From this pair again one allele can go, either capital Y or it can be small y. The capital R does not say that I will go only with capital Y. It's not like that. So this means they are showing something that is known as law of independent assortment. They are independently getting assorted, separated. So that is what you mean. So over here you see different type of phenotypes, the new phenotypes. That is what you denote by 9:3:3:1. You get it? It's because they are assorting or segregating independently, my dear children. So D becomes your answer for the question. Cool? So what is the answer? It's D. Okay? It's D. Now next question. A pedigree chart is given below. Identify the trait responsible for this inheritance pattern. Can you say what is it showing? Autosomal dominant trait, autosomal recessive trait, X-linked recessive trait, X-linked dominant trait. Now I'll tell one simple funda. Usually the questions are asked between these two things. The other two things are just given you as an option. Okay. Now, how do you find whether it is autosomal dominant or autosomal recessive? If the disease is found in every single generation. So, over here you see the parents are not diseased, but the children are. Over here their children are also diseased, right? So, in every single generation, if you are able to see, you call this disease as dominant one. But over here you see the parents are not diseased. You see them in children. Here also the parents are not diseased, you see it in children. That means it is not dominant, it is recessive disorder. And it has nothing to do with the sex of the organism. Over here they are equally affected. Okay, so it has nothing to do with the sex of the organism. So it is not X-linked recessive or X-linked dominant, my dear children. It is this particular disease which is not seen in every generation. You call this as autosomal recessive trait. I'm telling you again, if you find the disease in every single generation and you see them in at least one parent, then it will be autosomal dominant. If you do not see it in one parent and still the progeny is getting it, you will call it autosomal recessive trait. Okay? Cool? So yes, Aman, you will be getting the PDF for this as well, you don't need to worry. Fine. So we say, what is the answer for this? Ayyo, just a second. This is not a C, this is B. The answer for this is B. Cool? Chalo, next question. So there are few tips. If you want, you can just give a quick read to it. Yeah? Now, question number 4. Which of the following traits of a garden pea studied by Mendel was a dominant feature? So you know there is something dominant and there is something recessive. Dominant ones always express themselves. For example, capital T, small t, capital T is dominant, which is why the plant will be tall. So which one among this is a dominant feature? Just like tall, height is a dominant, this is the dominant feature in height. So which among them are the dominant feature? Any guesses, children? Come on, put it down in the live comment section over here. A, B, C, D. It is given in your NCERT textbook as well. So, terminal flower position, do you think it's a dominant character? No, children. Okay. In the case of pea plant, axial flower position is the dominant one. So, I will say this is recessive character. Green pod color as in if you have seen green peas, no, it has something like this, inside that you get peas. So this color that you see over here, the green color that you see over here, it is dominant character or recessive character? It is a dominant character. So I will say this is correct, but let's see the other options. Green color seed. So the pea that we eat is green in color, but is it a dominant character? No. Actually, it is yellow is the dominant character. So again, this is also wrong. We have to find the dominant one. White flower color. White is not dominant in the case of pea plant. It is purple which is dominant. So again, this is wrong. What do you have as the correct option? You have B as the correct option. Cool? Fine. Chalo. Next question. 4 children belong to same parents have the following blood groups A, B, AB and O. Hence the genotype of the 2 parents is. So you see the children have different type of blood groups. A hai, B is also there, AB is also there and O is there. How is that possible? You have to find the genotype of the parents. Now I'll tell you one thing. If O is there, always in the question if O is mentioned, just understand that O will be only seen if both the parents has O allele. That is IO should be there in both the parents because it is a recessive trait, my dear children. It is what? Recessive trait. So basically both the parents should have at least one O allele, then only it will be possible. So if A is there, if B is also there, AB, everything is there. So I'll say probably parent number 1's blood group is AO and parent number B's blood group is BO. This is the genotype. This is the genotype of parent number 1 and parent number 2. Now let's try to figure out the gametes. So in one gamete it can be A, in other gamete it can be O. Similarly over here one gamete can have B, other gamete can have O. When you fuse it, suppose the sperm from here, egg from here comes fuses, you get a child with AB blood group. From here it fuses, you get AO. That means this particular child, since A is there as a dominant character, so A will dominate and you will have A blood group. Okay. Now suppose O is there, B is there, you get BO. Again B is dominant over O, so this child will have B blood group. Over here O and O comes together. This is a recessive trait and hence it will express itself this time. There is no one to dominate this poor O. So you get O blood group. So what is the condition over here? This parent and this parent, are they homozygous? Do they have the same allele? No, they are heterozygous. Heterozygous as in the alleles are different. If it was AA, that means it is homozygous. If it was BB, that means it was homozygous. So both of them, they have heterozygous allele, my dear children. Okay, so both parents are homozygous A group. No, one parent is homozygous for A and another parent is heterozygous for B. No, we need both the parents to be heterozygous. One parent is heterozygous for A. Yes, it is heterozygous A, right? It is A blood group, but it is heterozygous A blood group. Similarly, other parent is what? Heterozygous B, my dear children. So this becomes the answer. Cool? Works? Chalo. If you got the answer, then tell me. Children, I don't see any interaction from your end, which is kind of saddening. Okay, you should be telling the answers up here, which is why I am here live, right? So over here, C is the answer. From next question, I believe that some of you will at least participate. I am not here to judge. No one will be judging you, even if you give wrong answers. Even I can be wrong at times. So please do not be worried about that. Just give me the answers. Yeah. So in the next question, it says that in a large number of crosses of 2 heterozygous round and yellow seeded pea plants. What do you mean by heterozygous? Heterozygous as in it's not the same. And round and yellow is also given. So probably the parents are like this. Heterozygous as in there will be a capital R and a small r. This is heterozygous. Both the alleles are different. Okay. Now yellow is also there. So I'll say this is the thing. This is heterozygous yellow. This is heterozygous round. So this is what? Round yellow parent. Now what is happening? In a large number of crosses of 2 heterozygous rounds, so you have similar type, one more parent. The fraction of offsprings that would probably appear wrinkled and green is, basically they are asking you about the dihybrid cross in which the ratio was 9:3:3:1. Always remember, I'll tell you a funda. 9 goes for the dominant character. Round is a dominant character. So basically 9 of them will be round and yellow is the dominant character between yellow and green. So 9 of them will be round and yellow. Okay. 1, which is the least number, it will be for the recessive trait. So over here what is recessive? Small r is recessive and R stands for wrinkled. R stands for wrinkled and Y stands for, the small y which is there, it stands for green. So wrinkled green is what? 1. Okay, what about 3 and 3? This is the new characters which were not found in the parent plant. So over here, this 3 stands for round. Initially round and yellow parents were there, no? Now we have to have new combination. This time round will be going with green, my dear children. Similarly over here, can you guess? Other 3 stands for what? the next 3 stands for what? It will be wrinkled and not green, but this time it will be yellow. Now what is the question? The fraction of offspring that would probably appear wrinkled and green, we have to find this particular fraction. So we see only one plant is like that among 16, right? So can I say 1 by 16 is the correct answer for this, right? If you remember this, you will be able to solve lots and lots of questions that comes around it. You get it? So this becomes your answer. We will say answer B is the correct one. So I was kind of expecting lots of you to be here talking to me, which is why I put so much efforts and put so much questions over here so that it will be interactive and things like that. Hi Jeet Pro, yeah. So I want you all to participate really well, otherwise I'll be really sad and I won't be coming for any live sessions from today onwards. Okay. So question number 7 says, garden peas selected for hybridization by Mendel were unisexual, either A or C, or bisexual or sterile plants. What do you mean by unisexual? That means there is only one, okay, that means there is only one sex in a plant. That is what you mean by unisexual. Bisexual means there are both the sexes, that is male reproductive part as well as female reproductive part in a plant. Now garden pea was chosen because it was bisexual. So Mendel did not have to go there and help with pollination, since the male flowers, with the male part which was there, it was releasing pollen, and the female part which was there, it was receiving it. Everything was sorted. So that is why he chose it. So I'll say it was bisexual, my dear children. Okay. It was bisexual. Cool. So this becomes your answer for the 7th question. C is the answer. Still I see no answers from your end. So Aishwarya ma'am is also here. What should I do, Aishwarya ma'am? We should be stopping doing this live sessions, right? Anyway, they are not answering. Probably, yeah. Do you want this?
Students, we are really taken a lot of like efforts in this. Okay, so we also have to stay late till evening. We are coming in the morning, and for you people to take this live session, we are going really like too late, and we are working really hard. But if you do not show this participation, we will be stopping this, as ma'am said. We will be just putting the recording videos as every time we do, right? But this is the first time we thought that we will come live and we will help you and we can talk with you, can solve your problems as well. But if this is the reaction we are getting response, then definitely we will be demotivated and we'll stop this. So if you are watching this, please put down your comments. You answer anything, whether it is correct, wrong, as ma'am told, doesn't matter. But yes, participation has to be there. Okay.
Jeet has now started answering.
Yes.
Really cool. That's the spirit, my boy. I hope I do not know the gender, but yes. I'm so sorry. Yeah. But anyways, okay. So he said or she said, which 3 scientists independently rediscovered Mendel's work? Yes, you are correct, my dear little child. It is what? De Vries, Correns and Schemark, who were the people behind it. They rediscovered Mendel's experiment. So Mendel was kind of sad that no one gave any bhav to my experiments. I did a lot of work. So then came these 3 people who said that we are going to discover your findings once more. Okay, so they did it independently, but they definitely did it. Okay, so yeah, Jeet Pro. Okay, cool. Great. So really kudos to you. So answer is what? D. Now the next question, in sickle cell anemia, glutamic acid is replaced by, this is an amino acid, my dear children. Instead of glutamic acid, what do you find there in sickle cell anemia? In sickle cell anemia, what is getting affected, my dear children? Hemoglobin is getting affected. Okay. In hemoglobin, you have a protein that is known as globin protein. This globin protein is getting affected because of some mutation in it. Okay, it is because one amino acid is getting separated, or it is totally different. Okay, so instead of glutamic acid, you have valine substituted over there, and if one amino acid is changed, the whole protein structure will obviously change. Yes, Jeet Pro. Yes, Bharadwaj. Great, my dear children. Good. I'm so happy. So it is valine, that is the correct answer. Always remember, G comes before V. So it was G before and then valine was substituted. Similarly, if I am asking you about the codons, which codon or which base pair is getting replaced, if you have the answer for that, you can comment down. Okay, chalo. The answer for this is anyways Ace. Kudos to you guys. Next question. Incomplete dominance in F2 generations has what? genotypic ratio equal to phenotypic ratio, genotypic ratio is 3:1, no segregation occur, phenotypic ratio is 3:1. What is the answer for that? Incomplete dominance in F2 generation. What do you mean by incomplete dominance is basically, for example, you have red colored plant, okay, and then you had white colored plant. Capital R is for red and small r basically is denoting white color, okay. G Pro is saying GUG is replaced by GAG. Exactly, that is the correct answer for the previous one. U which is there, okay, it was, wait. Initially it was GAG, then it was replaced by GUG. Okay, this is the, and this is why this particular codon is responsible for valine. That is the thing that happens. Always remember A comes before U. Okay. Chalo. So this was the condition where I was crossing red and white plants, right? Now over here the gametes, what do I have? Capital R. Over here small r. I make a cross. When I make a cross, all of the things that I get is like this. Now over here, what do you expect? What color it should be? Capital R is there, so it should dominate, right? But it does not completely dominate. Exactly, Jeet Pro, pink color is formed. What happens? This is incompletely dominant over the small r, which is why instead of giving red color, it is pink color. Now, you are asked about F2 generation. So what do you need to do? You need to cross them. So I cross two plants which are like this. What are the gametes? The gametes will be capital R, small r, capital R, small r. Now when I fuse them, what do I get? Capital R, capital R, capital R, small r, capital R, small r, and small r, small r. So over here, if I look for the ratio, the phenotypic ratio, as in how do they look? So this is red, both of them are pink, and this is what, children? This is white in color. Okay, so what I say, phenotypic ratio, how do they look? One is red, two are pink, and one is white. Now what is the genotypic ratio? What is the genetic makeup? So one is having two capital R, so I'll say it is like this. Two of them are having one capital R and one small r. How many? Two. Okay, and one of them is having small r, small r. So genotypic ratio is 1:2:1. Phenotypic ratio is also 1:2:1. So can I say genotypic ratio equal to phenotypic ratio? Yes. So this becomes the correct answer. Exactly, Jethro. Similarly, other children, Bharat, where are you? All the other children who are there watching this particular video, tell your friends also to watch this. Is really good way of revising things. Okay. So chalo, let's go on to the next question. Question number 11. How many possible sickle cell genotypes are there in the population? Any guesses? Now this is a tricky question, which is directly from your NCERT, but this is a tricky question. How many possible sickle cell genotypes are there in the population? Sickle cell anemia is caused because of mutations, right? So over here, this is the normal gene that we have, HBA. When you have two of them normally, it's fine. Suppose it's HBS, HBS, that means this person is affected with sickle cell anemia. Okay. Now when you have HBA and HBS, that means this person is a carrier. It's not affected, but it is a carrier. So how many genotypes do I actually see? 3. Now that is a tricky part, okay, that is a tricky part which is directly given in your NCERT, but it is not exactly explanatory, okay, which is why you have to remember it as such. Sickle cell genotypes, okay, even the normal is there, still we are including it. So you have 3 genotypes over here. As Jethro said, this is the correct answer, okay, answer is C. Going ahead to the next question that we have, in an ejaculation, semen consists of 10 lakh sperm. Okay, how many sperms are there? 10 lakh sperm. The number of sperms which contain Y chromosome is, how many sperm will be containing Y chromosome? So I know one thing, that in the sperm there is a nucleus, and the nucleus contains chromosome, that is both X and Y. So if 10 lakh sperm is there, so in how many number of sperms do you expect the Y chromosome? Obviously all the 10 lakh of them will be having a Y chromosome, which is why it is from a male body, right? So obviously it's not B, okay, this is not B, it will be 10 lakh itself. A is the correct answer for this. Okay, all the sperm might, Okay, okay, okay. I see a correction over here. Wait. Okay, okay, okay. I get it. Jeet Pro corrected me and my brain ignited. Yes, you are correct. Actually, it will be 5 lakh of them. Why 5 lakh? Because it is XY chromosome that is there in males, and 50% of them will be carrying X chromosome, 50% of them will be carrying Y chromosome. So it will be 5 lakh, not 10 lakh. Yes. So this becomes the answer. Cool? Chalo? Great, Jeet Pro. Yeah. So the answer will be not A, it will be B. That's a little correction from my end and I'm thanking you for that. Yeah. Chalo. Next. 13th. Hemophilia is more commonly seen in human males than human females. Why so? Why do you see hemophilia more commonly in human males than females? Hemophilia is what type of disease, my dear children? It is sex-linked recessive disorder. It is sex-linked recessive disorder. Okay? No, now if it is sex-linked recessive disorder, it will only show its effect when it is in recessive condition. So that means probably a person, okay, a person who is a female, okay, if she has one of her gene affected, still there is one normal gene to overshadow its effect, and hence this female would not be affected. Okay, she will be a carrier but she will not be affected. But in the case of male, they just have one X chromosome and the other one is Y. So anyways, if this particular X chromosome is affected, my dear children, there is no one to overshadow its effect. Right, this will show its effect and this guy will be what? Affected, which is why males are more likely to develop what? Hemophilia, or basically show up this particular character. So I'll say this is because the disease is X-linked recessive disorder. Always remember, okay? Hemophilia, color blindness, everything goes like this. Vishal says, thank you ma'am, so much, thank you so much. And Jeet Pro, really great. You're going really great. The answer is C. Cool? Now, the next question, which of the following assertion about alpha thalassemia is incorrect? You have to find what is incorrect. So, underline the term incorrect over there, okay? Now, alpha thalassemia involves HbA1 and HbA2 genes. Now, let me tell you, okay, these are the chromosomes. In this chromosomes, there will be HbA1 and HbA2. Similarly, you will have another pair, again, HbA1, HbA2. What is this chromosome pair number? It is, my dear children, 16th chromosome that we are talking about. Now suppose any of these genes are affected, that means you are having alpha thalassemia. Depending on how many genes are affected, you say minor or major thalassemia. You get it? All of them are going to code for what, say, alpha globin chain of hemoglobin. Hemoglobin is made up of 2 chains, alpha and beta. So the alpha globin is made with the help of these genes. So any of these genes are affected, that means that's a problem, right? So it involves HbA1 and A2 genes. This is correct. Is associated with the mutation or deletion of genes on chromosome 11. Chromosome 11 has nothing to do with alpha thalassemia. It is about beta. So I'll say this is the incorrect statement. But let's read the other options as well. Is associated with the mutation or deletion of genes on chromosome number 16? Yes, it is. Leads to reduction in synthesis of globin chain? Yes. Alpha globin chain is reduced, right? So which is the incorrect one? This is the incorrect one. Jeet Pro, again you are correct. Kudos to you, my child. Okay? So answer number B is the correct one. Going ahead with the 15th question, my dear children, others also please participate. If both parents are unaffected carriers for thalassemia, which is an autosomal recessive disorder, what are the chances of pregnancy resulting in an affected child? Now both of them are unaffected carriers, my dear children. They are just the carriers. So what do you expect? What are the chances of pregnancy resulting in affected child? Guesses? Any guesses? Okay, so it is a recessive disorder. Okay, so autosomal recessive disorder, that means it will be shown only under recessive condition. Only under recessive condition. So if there are 2 genes, suppose I'll say thalassemia over here. Okay. How do I explain this? Okay. They are carriers, that means, okay, this is the carrier gene, okay. Now we are crossing them. What do you get? When this fuses, you get HB, which is normal, it's all fine, okay. Now when this and this fuses, again you get HB and one HBr. This is again a carrier. This is not affected, they are carriers. Now when you fuse this with this, again you get HB and HBr. This is again a carrier, because you see it is recessive disease. So obviously you should be having two recessive genes together, then only this particular disease will be seen. Now you are fusing this and this, HBr, HBr. In this condition, you got both the recessive genes together. So what is the percentage of it? Only if you see 4 organisms are there, only one is affected. Can I say only 25% of them will be affected? Rest of them will be carriers or probably will be normal. Okay, or probably will be normal. So what are the chances of pregnancy resulting in an affected child? There will be only 25% chance of the child to be affected. So answer will be A. Okay. Ma'am, what about first PU revision classes? Okay, so we did Brahmastra first PU revision classes. We would be totally telling you about that really soon. Okay, once we are done with this particular lesson, we will let you know. We'll put down in the comment section, but we will definitely let you know. So don't be worried about that. Okay, cool. Chalo. Next question. What is the genotype of the person suffering from Klinefelter syndrome? What happens in Klinefelter's, my dear children? Over there, the person has to be a male, but somehow gets an extra X chromosome. So how many chromosomes do we have in our body? 46 of them. We have 46 chromosomes, my dear children. In that, 44 of them are autosomes, the one that does not define our sex, nothing. Then there are 2 chromosomes, that is X and Y, or probably X and X, that defines our sex. So if it is 44 autosomes plus XX, that will be a female. If 44 autosomes plus XY, it will be a male. But in the case of Klinefelter's, you get one extra X chromosome over here. This is the extra X chromosome over here. So what is the karyotype for this? As you can see over here, it will be 44A plus XXY. C will be the correct answer. This is the 16th question. Yes, exactly. Great, Jeet Pro. Cool. Now the next question. What percentage of children are colorblind if their father is colorblind and the mother is a carrier for color blindness? Okay, what type of disease are it, children? It is again sex-linked recessive disorder, right? So over here, what is the condition given? The father is colorblind. So the father is colorblind means there is only one X chromosome and that is affected. And the mother is what? Carrier of color blindness. That means she just have one allele which is affected. Now we have to find what percent of children are going to be colorblind, are going to be affected. They are not the carriers, they are going to be affected. Okay, let's see. So the gametes can be this, gametes can be this, gamete can be this from here, and gamete can be this from here. Let's fuse this and this. When you fuse it, you get X, XC. Now over here there is one to dominate, so hence she will be just a carrier. Now when I fuse this with this, I get X and Y. These are 2 normal alleles that you find over here. So this will be a normal boy. Okay, what about this and this? Don't you think both alleles are there? Obviously there is no one to dominate and overshadow the effect. So obviously this will be a colorblind child. Okay, then what about this and this? XC and Y. Again, Y is there, but Y does not have any effect over this. You need one other X chromosome to show its effect, right? So what can I say? This will be again a colorblind child. So I see 2 of them are not affected. One is carrier, but they are not affected. Okay, they are not colorblind, but 2 of them are. So can I say 50% chance is there? Okay, 50% chance is there. So the answer will be what? B. Jeet Pro, great my child, great, great, great. I see others also, 4 other people also in the chat. Why are you not telling the answers? I told you it's totally fine to be wrong. I don't know why I have written A over here. This will be B. Okay, cool. Chalo, next. In XO type of sex determination, where do you find XO type of sex determination, my dear children? Where do you find XO type of sex determination? If you remember, So in exotype of sex determination, females produce 2 different type of gametes, male produce 2 different type of gametes, females produce gametes with Y chromosome, male produce gametes with Y chromosome. What is the case? Exotype of sex determination is found in insects. Okay, for example, you find it in grasshoppers and stuff like that. Yeah. So over here, what is there? Suppose there will be one X chromosome and there won't be any other chromosome, which is why we say that is O. Okay, we denote it as O or 0 basically. From the female side, obviously it will be XX. Okay, now when you fuse them, what do you get? And this is O, that means there is nothing over here. There is nothing over here, so I'll just make it like this. When you fuse this and this, you get female. When you fuse this and this, you get one X chromosome and there is nothing over there. So this will be what? A male, my dear children. Again, when you fuse this and this, you get XX. When you fuse this and this, again something like this, which will again turn to be a male. So in exotype of sex determination, what is there? The males which are there, they are going to produce 2 type of gametes. One gamete which has the X chromosome, other gamete which does not have any other chromosome. You get it? So males produce 2 different type of gametes. That is the answer for this. It's not the females, it is the male. So I'm going to write female over here. I'm going to write male over here. Got it? And what do you call this condition as? Heterogamety. Okay, heterogamety as in 2 different type of gametes are being produced. Well, this is homogamety. Cool? Chalo. Next question. Jeet, great da. Okay. Haplodiploid sex determination is found in haplodiploid. Where do you find haplodiploid sex determination? Okay, that means suppose for example 16 chromosomes are there, 16 chromosomes are there, together it comes, you get 32 chromosomes, right? Suppose 16 chromosomes are there, okay, this is passed from mother's end and this is passed from father's end, you get 32 chromosomes. Similarly, from one side, okay, from one side you are getting 16 chromosome, from the other side you are not going to get anything. So what will be there in the progeny? 16 chromosome. So can I say it's half the amount of chromosomes? This is full. This is diploid condition. This is what? Diploid condition. This is half of 32, 16, right? So this is haploid condition. So what is this organism going to be, a male or a female? This is going to be female. So whenever that's diploid, it is going to be female. Whenever it is haploid, it is going to be male, my dear children. This is known as haplodiploid sex determination, and you find it in the very interesting honeybees. Okay, you find it in honeybees. D is the correct answer for this. Honeybees are totally unique. I hope you know that they have a unique system in their family. Workers, drones, queen bee, this bee, that bee and everything. So D becomes the answer for this. Cool? Next question. How is pleiotropy different from polygenic inheritance? What do you mean by pleiotropy? Pleiotropy is usually what happens is that there is one gene, okay, one gene codes for one protein. This is how things work, or basically I can say that one gene is responsible for one particular character. But in the case of pleiotropy, things are kind of different. There, one particular gene can have multiple effects on multiple character, my dear children. So that is what you mean by pleiotropy. Whereas polygyny, what do you mean by polygyny? I already told you one gene for one character, but over here multiple genes are there that will be responsible for one character, which is why you call as polygyny. So in case of skin color, multiple genes are going to define our skin color. That is an example. In pleiotropy, for example, one particular enzyme, when it is not there, it is going to affect 2 characters. That is what you mean by pleiotropy. Okay, so I'll say a single gene can exhibit multiple phenotypic expression. Is it correct? Pleiotropy means that only, right? A single gene is going to exhibit what? Multiple phenotypic expression, as in different characters are being affected because of that. That is what, how it is different from all the other ones. Cool. Chalo. So answer A is the correct one. I'm not going ahead with the other options because it is already correct. Yeah. Next question. Which of the following is not a symptom of phenylketonuria? Okay, so phenylketonuria is something that is shown. It is under pleiotropy itself. In phenylketonuria, what happens? So phenylalanine, we have it in our body. Okay, phenylalanine is converted to tyrosine with the help of an enzyme. With the help of an enzyme, that is phenylalanine hydroxylase. Okay, this enzyme is coded by one particular gene. and that gene function was to produce this enzyme and for this conversion. Now suppose this conversion is not happening because this enzyme is not being produced, that means this phenylalanine is going to get converted to phenylpyruvic acid. So it stays in our body and it causes mental retardation because it affects our brain and all. Okay, it affects our brain and it causes mental retardation. So I will say mental retardation is one particular symptom. So this is the correct thing. What about reduction in hair pigmentation, skin pigmentation, white urine? Is it correct? So let me tell you. I told you it's a pleiotropic trait. That means one gene is going to affect two characters. So one character is already affected. The second character is that since this enzyme is not there, tyrosine is not made, and when tyrosine is not made, melanin production is under scrutiny. Okay, it is not being produced very much, and when melanin is not being produced, your skin pigmentation, your hair pigmentation, it goes really less. Okay, you get hyperpigmentation, the color of it changes really bad. Okay, so can I say one gene is affecting two character? Yes, so it is an example of pleiotropy, and also reduction in hair pigmentation and skin pigmentation is a symptom of phenylketonuria. But white urine, this is wrong. This is a wrong option. So I will say C is the correct answer for this. Okay, what is the correct answer for this? C is the correct answer for this. This is not a symptom of phenylketonuria. Cool? Got it? Chalo, Jeet Pro again, great da. Next question. Others, my dear children, where are you all? What is the physical association of genes located on a chromosome called? Now this is easy, I want the answer from you all. Okay. What is the physical association of genes on a chromosome called? This is a chromosome, this is a gene, and this is a gene. What do you call this association as? Linkage, recombination, mutation, replication? Anyone? Tell in the... Okay, where are you? Over here. Chalo. Come on children, what kind of participation is this? Okay, Jeet Pro is saying A is the correct answer. Anyone is here to challenge? You should be challenging your teachers also. Initially Jeet Pro challenged me and look, he got the correct answer. So I am here for the challenge as well. So my dear children, the physical association is known as linkage. Okay, it is known as linkage. Now suppose they are far away from each other, so you call them loosely linked. If they are too close, you call them closely linked. Okay, so please remember this. This becomes your correct answer. Going ahead with the next question that we have over here, that is the 23rd question. What is the process of production of non-parental phenotypes known as? What do you mean by non-parental phenotypes, ma'am? Basically, okay, so you had round yellow plants and you have wrinkled green plants. You cross them. Now you are getting, so it was initially what? Round yellow and you have wrinkled green. These were the parental types. But in the progeny, when you are crossing them, you are getting a new type of plant itself in the second generation. What is it? What is it, my dear children? It is round green and wrinkled yellow. Ulta of this is happening. So why do you think this is happening? Okay, it's not just this. There are so many examples for it. Okay, why do we not exactly look like our parents? It's because probably the chromosomes which is there, it is mixing and matching. Some part of it is going there, some part of it is coming over here. So getting new characters, my dear children. And what do you call this as? You call this as recombination. Newer combinations are being formed. Yes, Jeet Pro. The answer is what? B. Recombination. Okay. So when the genes are located far away, they do much more recombination. Cool? So this becomes your answer for the 23rd question. 24th question. Which of the following is incorrect regarding ZW and ZZ type of sex determination? Ma'am, is this lecture enough for KCET? It will be really good for KCET as well, because you get it from your NCERT itself, right? The questions are being framed from your NCERT, so it will be really good practice for you all. And anyways, there are lots of concepts being cleared, right? So, yes. Cool? Chalo. So which of the following is incorrect regarding Z type of sex determination? First thing, tell me, where do you find this type of sex determination? You find it in birds. Okay. Now who has ZW and who has ZZ? Female or male? Tell me that. Who has ZW, my dear children? Female or male? And who has ZZ? chromosomes. These are chromosomes, okay? You just named it differently. Just like how we have XX and XY, similarly birds have ZW and ZZ. Any guesses? Who has ZW, female or male? Anyone who is watching this particular video, you can tell me. No one? That's kind of sad. ZW is female. Jitro has said, is it true? Do you think? Anyone there to challenge? Actually it is female. And ZZ will be what? Male, my dear children. For the first time, okay, the females are showing something that is known as heterogamety. What do you mean by heterogamety? That means it is going to form two type of gametes. In one gamete, it will be passing Z chromosome. In another gamete, it will be passing W chromosome. So this is known as female heterogamety. In birds, it is shown. But in the case of males, they are giving the same type of gametes. It is homozygous, homogametic condition. Okay, homogametic condition. In our case, in the case of human beings, sorry, I should not be writing there because the answers come up there. It is XX and XY. Here, who forms the different type of gametes? It's the male who form different type of gametes. So that is male heterogamety. So here, so this occurs in birds. It is correct. You have to find what incorrect statement. Okay, females are homogametic and males are heterogametic. Do you think females are homogametic? No. So this is wrong. This is the incorrect statement that we have over here. Aditya, ZZ male, great, great. Jeet Pro, great. Okay, now females are heterogametic and males are homogametic. That is true. Females are heterogametic and males are homogametic. Total number of chromosomes is same in both the male and the female. Arre baba. That is also true, no. So what is false over here? B is false, my dear children. Okay, so what do you say is the answer? It is B, close to B, and everyone who answered it. Okay. I see more number of people joining the class. So good to see that, children. Let's see, let's start a new life with the 25th question. So everyone who is here, you can answer, okay. The fact that every gamete formed has either of the alleles for a trait follows what? What do you mean by it? Ma'am, if you have this chromosome and this chromosome, this is capital R, this is small r, okay. In one gamete, you get capital R. In another gamete, you get this chromosome that is small r. What are you trying to show by this? What are you? Fact that each gamete formed has either of the allele, either of the both. You cannot have both the alleles together. Only one chromosome is going to go. That is for sure. So concept of unit factors, no. Law of dominance, no. Law of segregation, law of independent assortment. Tell me. What is the answer for this? Anyone over here in the class? Can you tell me the answer for this? Come on, children. What do you mean by that? So separation is happening. When separation is happening and one of them is going very sadly into one particular gamete, you say this is law of segregation. From a pair of alleles, only one allele will be going into one particular gamete. That is what is said by law of segregation. Always remember that. Okay? So C becomes your answer for the question. Exactly, Jeet Pro. Yeah? So I would love to remember your names also if you're there. Tell me, Mama there, give the attendance. I love to remember your names. We'll be talking often. Okay, if I can do any help for you guys, I'll be definitely doing it as well. Aditya Patil says D. Okay, for this answer? Okay, let me see. I hope you're talking about the 26th question. Okay, so which of the following blood groups is not possible in a child born to parents both having AB blood group? So if you are talking about this question, then you're absolutely correct, Aditya. It will be D option. Why D option? For the people who are new over here, what is not possible? What blood group is not possible? So over here AB is there. Anyhow, if you try to fuse it, A gamete, B gamete, A gamete, B gamete. If A and A fuses, you get AA. If A and B fuses, you get AB. If B and A fuses, you get AB. If B and B fuses, you get BB. This is A blood group. This is AB blood group. Again, it will be AB blood group. This will be B blood group. Do you see O blood group anywhere over here? No. So this becomes the answer. This is not possible. Great. Great, Aditya, and great, Jeet. Kudos to you, my dear children. Okay? So D is the answer for this. Cool. Now let's talk about the 27th question that we have over here. A dihybrid cross done following Mendel's hybridization experiment does not yield expected phenotypic ratio. Identify the law it is deviating from. Law of dominance, law of segregation, law of independent assortment. D says both A and B. So in a dihybrid cross, again I am telling you what do you mean by dihybrid cross. Two characters are being studied at the same time. For example, seed shape and seed color. Okay, so you crossed round yellow with wrinkled green. In the progeny you got a ratio 9:3:3:1. So what law is it actually deviating from? Does not yield the expected phenotypic ratio. Can you suggest? Identify the law it is deviating from. Any guesses? Jeet Pro says Ace. Others, do you have an answer for this question? 9:3:3:1 he is getting. That means you are getting new parental type, new non-parental type as well. This will be round and yellow, sorry, round and green, and this will be wrinkled and yellow. Aditya, Patil and Jeet Pro, answer for this. Anyone else who has the answer for this? A dihybrid cross done following Mendel's hybridization experiment does not yield the expected phenotypic ratio. Identify the law it is deviating from. Or can you just say identify the law it is showing? Can you say that? Identify the law it is showing. That will be the more correct option for this. It will be C, law of independent assortment, where both the alleles are assorting independently, my dear children. They are assorting independently, which is why you get newer combination. If they were not assorting independently, you will just have the parental combination. Since they are saying I do not care whether capital R goes with capital Y or capital R goes with small y, which is why you are getting newer combination. Okay, we get round and green plant. You get it? So this becomes the correct answer if the question was like this. Okay, if the question was like this. Cool. Shagufta says C. Yeah, great. Okay, try this question, all of you guys. 28th. Which of the following cannot be the test cross ratio? Which of the following cannot be the test cross ratio? 1:1, 1:2:1, 3:1, both B and C. What do you mean by test cross? Test cross is basically when, okay, I am not sure about the genotype of the parent plant. I just know, for example, it is tall. I don't know whether it is tall like this or tall like this. So what do? I do a test cross where I cross these plants with a dwarf variety, pure dwarf variety. For example, I am going to cross capital T small t with small t small t. So what is the ratio which I cannot get over here? That is the question. Sumit Jabagon, can you please tell me what you did not get? I am ready to re-explain it, my child. Okay, which question are you not getting? Can you please tell me? So over here, let me get to it. Okay, so when I cross, I get capital T, small t, small t, small t. When I cross it, capital T, small t, capital T, small t, okay, and then small t, and I get small t, small t. So I see 1:1 ratio. Both of them are capital T, small t, and over here small t, small t. So it is 1:1. That is entirely correct. Okay. Now one more option is there where the things are like this, where I am crossing this with this. I see. Sumit, please tell me which is the thing that you are not understanding. I will be definitely responding to it. Aditya Patil says A for this answer. Cannot be a test cross ratio. That is the question. So when I cross, again, this will be all of them if you try to cross it, my dear children. All of them if you try to cross it, they all will be like this. So can I say this and this, both of them cannot be a test cross ratio, because anyway you are getting 1:1 in a test cross ratio. This cannot be the test cross ratio. So both B and C are the one that are not correct, right? So the option will be what? The option will be D. Both B and C you cannot get in the test cross ratio. So option will be D. So whoever got it as D, you are correct. So please read this question really carefully. Even I got confused for a second. Okay, which of the following cannot be the test cross ratio? This cannot be these two cannot be the test cross ratio. This can be 1:1. You got it in the test cross, so this can be a ratio, but these cannot be. Cool. So your answers become D, not A. Okay, it's D. Next, a patient goes to the doctor worried about a simple cut, that the simple cut is taking unusually long time to clot. The doctor identified the patient's condition as, what is the patient condition? For example, you got a cut and it keeps on bleeding. Okay, it keeps on bleeding, it's not stopping. What is this condition? Sickle cell anemia? Do you think it is that? Or thalassemia, or phenylketonuria, or hemophilia? I'll give a clue. Okay, England's Queen Elizabeth have this particular disease. This particular disease. This is sex-linked recessive disorder. Okay, this is sex-linked recessive disorder. Can you tell which is it? Shagufta says D. Jeet Pro says D. Yes, it is hemophilia, my dear children. In hemophilia, what is happening? The protein that is responsible for clotting, it is mutated, okay, it is not going to be formed, which is why you do not get clots. The blood keeps on going. It is hemophilia, okay. So this becomes your answer. 29th is done. Let's move on to the 30th question, and now we have 10 more questions and that's all. Okay, so please, we want active participation. First question, so 30th question. Which of the following is not possible in marriage between carrier woman and normal visional man? Carrier woman, you're talking about color blindness. So, woman is carrier, that means only one chromosome is affected. And normal visional man, as in the guy which is there, he has normal vision, there is no chromosome that is affected. And now you are asked, what is not possible? Okay, what is not possible? So over here, let's fuse X, XC, X and Y. When you cross this with this, you get XX. This is what? A female, and she does not have any alleles which is affected. So I'll say it is a normal female. So I can get normal female. Female, oh my god, yeah. Now X and Y. This is again, none of them are affected, so I will say it is a normal male that I have over here. XC and X. So over here, one allele is affected and this is a female. So can I say this is going to be a carrier female? She is not affected, she is not color blind because the other one is there to overshadow the effect. She is just a carrier. Okay. Now XC and Y is there. XC and Y. Is there any other X to overshadow its effect? No, my dear children. So this is a guy, this will be a male, and this male will be color blind. There is no option to be a carrier in the case of male. Okay, so you are getting normal female, normal male, carrier female, color blind male. So which is not possible? Color blind boys are possible, you saw it. Carrier girl is also possible. Color blind girl, do you think color blind girl is possible? No, none of them are color blind. So this is not possible. Normal boys and girls, they are also possible. So this is not possible. You have to make a cross, then only you can show. Oh, okay. How to video going live by same teacher. So that is a premiere that has been done. Okay, this is live. You can, Siddharth, I can actually see, see your comments. So you can trust me over here. This is a live. Okay. So color blind girls is the answer, my dear children. Answer is C. Cool? Works? Aditya says C, Shagufta says C, Jeetro says C. Great. Cool, 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 cool. Really good. 31st question. Tall stature, underdeveloped testis and gynecomastia are found in which disease? This is symptoms of which disease? Yes, you can tell me the whole answer and like that. It will be really, really good. Tall stature as in they will be really tall. Testis is underdeveloped as in probably they are not producing enough amount of sperms. My dear children, probably they are not producing sperms itself. Okay. And gynecomastia as in development of breast. So if it was happening in females, it was all normal, but this is underdeveloped testis, gynecomastia, tall stature. This means there's a problem over here. This is a disorder. And what is this disorder? Exactly, Siddharth, exactly, Tanay, great, Shagufta, Jeet Pro, everyone is correct over here. Kudos, my dear children. Now I am happy seeing every one of you attempting the answers, attempting the question. Aditya, yes. So it is Klinefelter syndrome where there is one extra chromosome. It had to be XY, but there is one extra chromosome. It says, I also want to be there, and that causes whole trouble. So this becomes your answer. Cool. Kudos to everyone who got it correct. Next question. Chalo. A man marries a woman and both do not show any apparent traits of inherited disease. Okay. So man and woman both are not showing any disease character. 5 sons and 2 daughters are born. How many? 5 sons and 2 daughters, my dear children. Okay? 3 of their sons, among the 5, 3 of them are having disease. That's sad. However, none of the daughters are affected. Daughters are not affected. The following mode of inheritance for the disease is what? Sex-linked recessive, sex-linked dominant, autosomal recessive. What is the answer for this? Tell me. One thing I'll tell you, one clue I will give you, okay? Whenever you see the males are getting affected and the females are not, okay, very much, just understand it has to be a recessive disorder and it has to be sex linked as well. What happens is that suppose XX chromosome is there in the case of females. If one chromosome of it is affected, the other one says that I am here to protect you, I'll save you from everything. So this particular female will not show any disease, she will be just a carrier. But if the same thing happens with a male, if one chromosome is affected and the other one is not, still there won't be anyone to overshadow its effect, there is no other X chromosome. So this guy will be obviously diseased, my dear children. This guy is obviously going to get disease. So which is why males are going to get disease. So it's an X-linked recessive disorder. That means males are going to get affected the most. The females, the daughters which are there, they're not showing the disease. Probably they might be just a carrier. They might be just carriers. Okay. Can you guess the genotype of the woman and the man who are there in this particular scenario? Can you guess the genotype, my dear children? Suppose it is color blindness. Okay. What will be the genotype? The male which is there and the female which is there. What is the genotype of the male? Anyone? So Aditya, Jeet Pro, Shagufta, Tanay. Tanay says D for this, autosomal recessive. No, my dear child, it is not autosomal recessive. It is not autosomal recessive, okay? Just check through once more. Just go through the question once more. Shagufta says A, Aditya says A, but anyone, can you tell me the genotype? The genetic makeup of the male over here with this particular scenario, can you tell me? Okay, XY, but is it, okay, normal XY, okay. Jeet says X-Y, probably you are saying that is affected. Okay, anyways. Female over here, do you think she will be affected? If it is affected, can you show like this? Can you write like this if you think it is affected? Okay, let me break the suspense for you. Man marries a woman and both do not show any apparent traits of inherited disease. Okay, so I'll say if I assume that this condition is happening, I already got the answer, that is a sex-linked recessive disorder. Okay, that means the female probably would have been a carrier, my dear children. The male is normal, which is why male is anyway not showing the effect. He is not diseased. The female is a carrier, she is also not diseased, she is just a carrier. Now, when you fuse them, what happens? X chromosome, Y chromosome, X chromosome, XC chromosome. When you fuse it, you get XS, normal female. Okay, when you fuse this and this, again, normal female, that's a carrier female, but she is not affected. Okay, when you fuse this and this, XY you get, that is a normal male, but when you fuse this and this, you get a diseased male up over here. So you get it. So which is why males are affected. None of the females over here you see are affected. They are carriers, but they are not affected. So this is the exact condition that is happening over here. You get it? Shagufta, yes, my dear child. Okay, so this will be the genotype. Always remember, questions can be framed like that also. So A is the correct answer, as you all guessed, but you should be knowing the genotypes also. Okay, so always remember, if it's said that man is not having any disease, it has to be like this, and the female will be definitely a carrier if the son has it. Cool. Let's go to the next question. A pleiotropic gene is what? What do you mean by pleiotropic gene? Controls a character only in a combination with another gene, controls multiple traits in an individual, is expressed only in primitive plants, is a gene evolved during Pliocene. What is the answer for this? Someone sent me heart and someone is scared. Oh my god. Thank you, thank you, thank you. Well, listen to me, my dear children. Pleiotropic genes mean, I already told you, usually we say one gene for one character. But pleiotropic genes are the ones where one gene will be responsible for not one character but multiple character, and multiple character means multiple phenotype. So control a trait only in combination with another gene, wrong. Controls multiple traits in an individual, yes. So this becomes the answer. Example, great Shagufta, great Siddharth, great Aditya, kudos. Okay, so answer will be B. Chalo, 33rd question that we have. Match the terms in column 1 with the description in column 2 and choose the correct option. Now that is a question that I took from NEET, okay. You can find it anywhere, it's an easy question, it's a very easy question that I gave you. Dominance, what do you mean by dominance? Dominance means one, in a pair of allele, one is showing its effect. That is what you mean by dominance, right? But it has to be different, both the alleles have to be really different. Shagufta said phenylketonuria, yeah exactly, phenylketonuria was the example for the other pleiotropic gene, okay. Chalo, Aditya Patil, Sujay Darbi, there's a new person over here, hi. So yes, let's see. So dominance goes with, many genes govern a single character, okay, let's go from here. Many genes govern a single character, which will be the answer for this? Polygyny. Multiple genes controlling a single character, you call this as polygyny. Poly means many and genes mean gene, you know, right? In a heterozygous organism, only one allele express itself. This is what? Dominance. In heterozygous, as in this condition, one gene is showing its character. That is this gene, and that is what? Tall. It's showing its effect. So this is dominance, my dear children. So I'll say A goes with 2. A goes with 2, let's see the option. A goes with 2, you just have two options like that, let's cut these two options, okay. Codominance, codominance means both of the genes are saying that I will be expressing myself. For example, in the case of AB blood group, A allele, B allele, both of them say both of us will be showing our effect. This is known as codominance. Both of them are equally showing their effect. It is codominance, my dear children. So in a heterozygous organism, both allele express themselves fully. This is known as codominance. So B goes with 3. Let's see, B goes with 3, that is happening in option number option number D, so I'll cut this also off. But let's see the other options also. Pleiotropy, one gene having multiple effects. Single gene inheritance influences multiple character, that means C goes with 4. C is going with 4 and D is going with 1, this becomes your answer, okay. Tanay says it's D, yes, it's D, it's not C, how can it be C? Guys. Make the correction over there. How can it be C? C says, okay, if you're not able to see the option because I did lots of chiturputur over here, I can show you the option. C says A is with 2. A is with 2, that is correct. And B is with 1. Codominance with 1, nah, no, that is not true, right? So this is the correct option. You have D as the correct option, okay. Oh, yo, my options are always next level options, my dear children. So your teacher probably has some problem, which is why the options are like that. Anyways, you got the answer, I got it, I told you correctly, okay. So just typo error, please forgive me. 35th question, a plant having the genotype A, capital A, small a, capital B, small b, will produce dash many kinds of gametes. Can you tell me the answer for this? Now this is not given in your NCERT. But let me tell you, all these questions that we have taken, some of it might be from NEET category also, some of it is from the government's MCQ list that they have given, the question paper list that they have given. So you have to tell me. So this is from that list. So you can expect a question like this. B. Okay, Siddharth and Jeet Pro says B. Let's see. How do you calculate it? Remember, always remember this trick, okay? How many heterozygotic condition you have? You have two conditions like that. Heterozygote as in, for example, over here, heterogametic as in, you have one allele, another allele for the same. So they are heterozygous. Over here also, one allele, another allele. Okay. How many of them? This is 1, this is 2, where this is happening. 2 of them are there. So always remember this particular formula, 2 by n. How many heterozygotes are there? You put that n over here. Here there are 2, so I will raise 2 raised to the power 2. So 2 raised to the power 2 means what? 4, my dear children. So your answer will be 4. 4 gametes will be formed over here. Okay? Yes, 2 power n formula. Oh my god. Oof. I am truly happy seeing this. Great. Okay. So now a question for you out of this. Okay. If it was like this, how many gametes are there in this condition? You can write 35B. Okay, 35B. This is the answer for 35B. Tell me for the answer for this. How many gametes is there in this particular condition? Being a NEET student, it is like revision for me and very helpful to correct our mistakes in life. Great, yes, Jitro, truly happy to see that. Children, also do not forget to like, share and subscribe, okay? I forgot to say that because I was in hang of teaching and obviously I love doing that. So yes. So over here also you will be finding 4 gametes only because we just have to find the heterozygote number. So only both of them over here, this is heterozygous condition, this is also heterozygous condition. So still it's 2 only. So 2 raised to the power 2 itself, it will be still 4. This is homozygous, right? C is the same thing. They are not heterozygous. Aditya, your teaching is amazing. Oh, thank you so much. Means a lot. Chalo. So B is the answer, okay? 36th question, my dear children. Inheritance of skin color in humans is an example of point mutation, polygenic inheritance, codominance, chromosomal aberration. Can you tell me? Skin color, my dear children, I already gave you an example. Skin color, no, it is controlled by usually one gene, one character, right? But over here, so many genes are responsible for one particular character, that is skin color. What do you call this condition as? This is known as polygenic inheritance. Too many genes controlling a character is known as polygenic inheritance. Okay, cool. So your answer will be this. Remember like that only. Polygenic inheritance, example skin color. Pleiotropy, example phenylketonuria. This should be must do topics in your head. Okay, it should be associated like that. Shagufta, Tanay, Aditya, Jeet Pro, everyone got your answers correct. Great. Awesome. Okay. Ma'am, is there any poll system? No, there is no poll system over here. We look forward to it from the next session onwards if we can do something about it, okay? Next, we have 37th question. Select the correct statement from ones given below with respect to dihybrid cross. Chalo, let's solve it. Tightly linked genes on the same chromosome shows higher recombinations, okay? Genes far apart on the same chromosome show very few recombination. Gene loosely linked on the same chromosome shows similar recombination. Tightly linked genes on the same chromosome show very few recombinations. What is the answer for this? Any guesses? So I'll explain something because I'm seeing this question from a long time and I know it's kind of confusing for at least a few of you. Listen to me carefully, okay? This is a chromosome. This is another chromosome. Okay? So you have a gene over here and a gene right over here as well. I hope it's visible. Okay. Suppose this part of the chromosome is cut and now is transferred to this particular site. Okay. So I am just changing the colors for you to understand this better. Initially over here it was like this and it was like this, okay? So what will be the new combination that you find? Ayo, just a second. Just give me a second, my dear children. The new combination that I will be finding will be something like this. One more change over here. I have to add something like this. Okay. So this is another gene, this is another gene, these all are other genes. Okay. Now when I do the whole change up over here, lots of things are going to change. Lots of the things are going to change. So the upper part remains the same. The lower part, if you see, they have exchanged themselves. So now what do you get? You see this has come this side and this guy and this guy have moved that side. Don't you see a new chromosome over here? This chromosome and this gene and this gene were on the same chromosome, but now it's not like that. You find a new combination up over here. This is known as recombination. Now these two guys are always together. These two guys are always together. Here also they have come just like that. Okay. Okay. So can I say that the one which are very close are known as tightly linked genes? They are tightly linked genes, and wherever they go, they will go together. So do they show recombination? Do they show new type of combinations, my dear children? No. They are very close, so they have to move together. They have to move together. Okay. So they show less recombination. They form very less recombination. But what about this gene and this gene? This gene is there, this gene is there. This guy moved that side, the other guy came over here. So can I say they are loosely linked? These guys which are over here, they are loosely linked, and hence they show higher amount of recombination. They show higher amount of recombination. So what will be the correct statement? Tightly linked genes on the same chromosome show higher recombination. Do you think so? They are tightly linked. Are they showing higher recombination? No, this is wrong. Genes far apart on the same chromosome show very few recombinations. Genes which are far apart, do they show very few recombination? In that case, it should not have been changed, but it's changed now. So this is also wrong. Genes loosely linked on the same chromosome show similar recombination. What do you mean by similar recombination? Nothing like that. Tightly linked genes on the same chromosome show very few recombination. Yes, they show very few recombination. There also they are going together only. So you are not getting a new combination up there. You get it? So your answer will be what? D, my dear children. So everyone who said D as the correct answer, you got it correct. Siddharth, Shagufta, great. Great, my dear children. So D is the correct answer. Always remember the genes which are close, they show less amount of recombination. Okay? Aditya, great, great, great. Can we move on to the next question? 38th question that we are having up over here. Who noted that the behavior of chromosomes were parallel to the behavior of genes? Now this is a straight question. Can anyone guess? Who noted that the behavior of chromosomes were parallel to the behavior of genes? T.H. Morgan, Gregor J. Mendel, Alfred Stuyvesant, and Walter Sutton and Theodore Boveri. It's a very straight, so it will be D over here, my dear children. Okay, these guys were working and they said that whenever you're talking about chromosomes and all, please understand that chromosomes are the one that has genes. So obviously when chromosomes move, genes are also moving. So basically the behavior of chromosomes was parallel or basically same like the behavior of genes. So this was said by Walter Sutton and Theodore Boveri. Okay. 38D, Shagufta, great. Aditya, great. Siddharth, great. Jeet Proda, just check once more. Okay. So answer is D. Now next, 39th question. In monohybrid cross between heterozygous individuals, percentage of pure homozygous individuals obtained in F1 generation is? Monohybrid cross between two heterozygous individuals. Heterozygous means, for example, capital T, small t, capital T, small t. How many of them will be small t, small t? That is the question. Percentage of pure homozygous individuals. Sorry. How many of them is capital T, sorry, small t and also capital T, capital T? What is the percentage of them? Let's cross. The gametes can be capital T, small t, again capital T, small t. When you fuse this with this, you get capital T, capital T. This with this, you get capital T, small t. This with this, you get capital T, small t. This with this, you get small t, small t. So here you get homozygous condition, both the alleles are same. Here also homozygous condition, both the alleles are same. So 25%, 25% together becomes what? 50% of them will be homozygous, pure homozygous. Exactly. Great. 39B is said by Shagufta. Siddharth said D. Checked. How D? Jeet Pro 50%, great, and Aditya also 50%. So this becomes the answer. Again I have written A. Oh my god. What was the thing that I was thinking? Okay. Okay. Chalo. Next. 40. Last question. Okay, but this we are going to end. Ashura ma'am is also waiting. She didn't have dinner probably. She's yawning over here. My class is that boring. I didn't know that. So, yes. In the F2 generation of Mendelian dihybrid cross, the number of phenotypes and genotypes are. This is the last question. Very easy question. Mendelian dihybrid cross, the number of phenotypes and genotypes are. F2 generation, my dear children. So, F2 generation. So, round, yellow and green, wrinkled and green. Okay, wait. Very straight question, but for children who do not understand it, that's why I am explaining you. You had two parents. When you fuse them, you got round and yellow, one. You got wrinkled and green also. You also got round and green. This is the third phenotype. Phenotype as in how do they look, okay? And then wrinkled and green was there, so I will say yellow is there now, the new thing. So this is the fourth phenotype. So you see four phenotypes, four physical differences or four characters that you can see. Genotypes you cannot write over here, there are so many. How many of them are there? 16 genotypes will be there. 16 genotypes will be there, okay? 9 of them with round yellow, okay? 1 with wrinkled green, 3 of them with round green and 3 of them with wrinkled yellow. That is 9:3:3:1 ratio. 9 the parental dominant, 1 the parental recessive. These are the two new guys that you saw over here. So 16 genotypes are possible and 4. So I will say this is the answer. So you 40D. Did I in F2 generation of dihybrid cross the number of phenotypes and genotypes? Hey, how it is 9? How it is D, da? How it is D? How can 40 be D? Someone. It will be this A, not D. Okay, you are talking about dihybrid cross, right? So, the total they are asking you. So, this is the answer. Okay, 9 plus 3, 10, 11, 12, 13, 14, 15 and 1, 16. So, this will be the thing. Cool? Anyone who has a confusion in that, do let me know. So, with this, my dear children, we are ending our session. Okay, so Aishwarya ma'am is asking, hello students, how was the session? Did you enjoy solving questions with Jasmine ma'am? I hope you did. Well, if you did not, please let me know. Okay, I will be gladly improving myself. And it was the first live that I had after a really long time. So I was kind of really scared, okay, to be there in front of you guys. But yes, you made me so comfortable, so I'm kind of really happy, my dear children. But definitely do let me know if you have any doubts in the comment section, and also show me your love in the comment section. Would be seeing it later on. So it's time to go have dinner and also have a good amount of sleep. So you also please make sure you're sleeping in a nice way, and also you're having proper food and enjoying your life as well. So that's all. Shagufta, ma'am, your teaching is fabulous. Thank you so much. Means a lot to me, the world to me, and very helpful. Thank you. Thank you so much, Aditya. So, my dear children, 39 asked in F1, so 100% dominant. 39 now. Beta. In a monohybrid cross between two heterozygous individual, percentage of pure homozygous individuals obtained in F1 generation. Listen, Over here, they have given the parents thing, no? This is not the normal monohybrid cross that you're talking about. The normal monohybrid cross is between pure tall and pure dwarf. But here they have given, it is two heterozygous individual that they are crossing. Now they are asking you, what is the F1 generation, the first generation that you get over here. Okay, so that is why it will be this. The answer will be this. I hope you got it. Okay, I hope Siddhartha, it's clear to you. And 40th question you want me to explain once more? Okay, please explain the last question. Okay, fine, I'll do that. Phenotype you are asked. So round and yellow is one type of character, wrinkled and green another type of character, round and green other type of character, and wrinkled and yellow another type of character. So how many characters you got? 4. So can I say 4 phenotypes are there? Yes. About the genotypes that you have, the genetic makeup that you have, when Mendel did his experiment and you did the Punnett square, you crossed every single thing that you got over here in the form of gametes and stuff like that. Okay, so there were 16 different genotypes observed. How many? 16 different genotypes. Okay, that is why we put 9 of them, 9 of the genotypes showed red, round and yellow. Now the genotype of round and yellow can be like this, round, yellow like this, could be like this also, you get it, or can be like this, but there were so many. How many? 9 of them coming in either of these category. 9 of them were round and yellow, and 3 of them were round and green, 1 of them was wrinkled and green, and 3 of them were wrinkled and yellow. So how many genotypes did you have? 9, 3, 9 plus 3, 10, 11, 12, again 3, 13, 14, 15 and 1, 16. So you had 16 genotypes. Got it? It is a total genotype that they are asking you. Total. You just have to calculate the ratio 9:3:3:1 and tell the answer. That's all. Simple. Okay, do not make it complicated. It's a simple question. Cool? Do not take it to NEET level. It will be complicated for you then. If there is any connection from my end, my dear children, after this class, I will just go through, I will correct and I will let you know in the comment section. Okay? So I think that's all. Yeah? Shagupta says make a session like this, molecular base of inheritance, definitely doing that. Okay? We will be definitely putting for every chapter. So I think that's all, my dear children. I will sum up. Good night for today and bye-bye. Take care. Cool. So ma'am, shall I end it from here? Yeah.